Hello बच्चों, let's see the question. So the question says restriction enzymes. So हमें इन चार ऑप्शंस में से आइडेंटिफाई करना है कौन सा स्टेटमेंट restriction enzymes को बेस्ट डिस्क्राइब कर रहा है. So the key concept in this question is to understand restriction enzymes. So restriction enzymes are firstly types of endonuclease enzymes, and therefore they are also known as restriction endonucleases. So endonucleases are enzymes which can cleave DNA at the middle region of the nucleic acid. And restriction endonucleases are specific types of endonucleases that can cleave DNA at a very specific site. So the restriction endonuclease enzymes first recognizes a sequence of nucleotides present in the DNA, and this sequence of nucleotide which is recognized by the enzyme is the recognition site. So once the restriction enzyme recognizes the recognition site, then the restriction endonuclease can cleave the DNA into two fragments at a very specific site. So restriction endonucleases were first discovered in bacteria. So bacteria uses this enzyme to fight against bacteriophage infection. So the restriction endonucleases present in the bacteria are very specific for the viral DNA only, and therefore can cleave the viral DNA into two fragments, rendering them ineffective. So the restriction endonucleases cleave the viral DNA, and the cleaving of the viral DNA results in preventing bacteriophage infection. So now if we see one example of a type of restriction enzyme, so EcoR1 is a type of restriction enzyme which is derived from the bacteria E. coli. Now this enzyme recognizes a sequence that is G A A T T C in the 5' to 3' direction, and the complementary strand C T T A A G. So the sequence which is recognized, so this is the recognition site for EcoR1. So the EcoR1 can recognize this nucleotide sequence present in DNA, and then it cleaves the DNA at this point and this point. So due to the specific cleavage by the enzyme EcoR1, we will get two fragments of DNA as follows. So the cleavage at this specific site will result in the formation of two cleaved strands of DNA. So these are the two strands of DNA which will be formed due to the cleavage by the restriction enzyme, that is EcoR1. So now if we come back to the question about the restriction enzymes, then the first option says that restriction enzymes are endonucleases which cleave DNA at specific sites. So this is the correct answer, because we saw that the restriction enzymes are responsible for cleaving DNA at specific sites only. Now, the second point says that they can make complementary DNA strands to an existing DNA or an RNA. So, this is not the correct statement because complementary DNA is formed by DNA polymerase. Then the third option says cut or join DNA fragments. So the enzyme which is responsible for joining DNA fragments is DNA ligase. So again, this won't be the answer. And the fourth is are required in vectorless gene transfer. So this option is also incorrect because restriction enzymes are used in vector gene transfer. So these enzymes are used when we require a vector for gene transfer. So in a vectorless direct gene transfer, we do not require restriction enzymes. So the correct answer for this question is option number 1. So हमारा final answer है option 1, that is restriction enzymes are endonucleases that cleave DNA at specific sites. So ये हमारा final answer है. Hope you understood this well. Best of luck. Thank you.